Hello, and welcome to our continuing discussion on the cardiovascular system. At this point, we're touching on blood vessels. We've already discussed the blood that those vessels carry, and we've already discussed that heart that pumps around that blood. So here we see how blood vessels form a closed circuit. And we're going to go over arteries and veins and some other related items. So as a general overview, we can classify all these vessels in the human body under three main categories. We can call them arteries, and by definition, they are going to carry blood away from the heart. And another general category we can talk about are capillaries, and those are very small and microscopic blood vessels that run between venules and arterioles. And lastly, veins are going to be blood vessels carrying blood back towards the heart. And then, generally speaking, these arteries are typically carrying oxygenated blood, and the veins are typically carrying deoxygenated blood. And then the capillaries, generally speaking, are going to be a site of exchange. And at the capillaries, this is where perfusion is going to occur. This is where oxygen gets dropped off the important organs and carbon dioxide picked up as well. And we might also be picking up other things like cellular wastes such as urea. In this diagram on looker's right is a sort of a museum specimen where they've injected a sort of plastic into the blood vessels and then corroded and washed away, so to speak, all the other tissues, leaving behind a sort of print of the cardiovascular system. So let's look at the structure of the walls of a blood vessel. And we have three main tunics that surround it. Now, before we get into that, let's go ahead and discuss this histology image. Notice in this histology image how the artery tends to be more round and also is going to have a little thicker middle portion there, whereas this vein tends to be a little bit more flattened and collapsed at some times. We're going to discuss some subtle differences between arteries and veins in a short few. On the lower left-hand corner, keep in mind that these blood vessels we're talking about are on the organ level. So that means these organs are made up of a variety of tissues. So we're going to see epithelial tissue that surrounds the inside of the lumen of the blood vessel, and then the connective tissue might be covering the outside of this blood vessel, providing a basement membrane. And then we also have smooth muscle tissue that's going to help that vessel constrict and dilate. So it's an interesting organ how we have these multiple tissues all coming together to provide something simple and basic. So we have three tunics that surround the lumen. When we say lumen, we're talking about the sort of empty space inside the blood vessel. Perhaps a good way to think about these tunic layers are to think of the root word of a tunic. That's an older term that might be described a sort of cloak that might cover a person. So just like a person may be robed in a tunic, here the blood vessels are having multiple robes around them. So let's take a look at each of these. In this diagram, notice on looker's left we have an artery, and on the right we have the vein, and connected between the artery and the vein are the capillaries. Now let's take a closer look at these tunics, these layers of vessel walls that surround the lumen. We'll start out on the top with the innermost layer, that is the tunica intima. And that's going to be composed of a layer of endothelium over a thin subendothelial layer. And that subendothelial layer is going to be made up of areolar connective tissue. This nice thin endothelium allows to create a smooth, frictionless surface that blood can move swiftly past. It is noteworthy that this tunica intima does become continuous with the endocardium of the heart. And then we move on to the tunica media. That's going to be our middle tunic, and that's composed of smooth muscle tissue supported by elastic fibers. We can then, thanks to this nice thick layer of smooth muscle, this tunica media results in vasoconstriction. When we say vasoconstriction, we're talking about a constriction of those blood vessels. And then when we relax this smooth muscle, that's going to result in vasodilation, opening or increased diameter of the lumen, so to speak. And then we get to our third outermost layer, the tunica externa. That's going to be composed primarily of areolar connective tissue with some elastic and collagen fibers mixed in. This tunica externa is going to help anchor this blood vessel to other structures, and also you can see there's even smaller vasculature in there that's going to provide nourishment to the blood vessel as well. And in between this artery and the vein, we see the capillary network. Again, this is where we're perfusing tissues. Keep in mind that we are on a cellular level. Some of these capillaries shown here on a cellular level. Notice the presence of the nucleus. The red blood cell is just about the diameter of the lumen of this capillary. So generally speaking, these arteries tend to have a thick tunica media and a more narrow lumen. They're also going to be more elastic with collagen fibers. And again, these are going to be more elastic than the veins. And that's because they have to absorb a sort of recoil from the heart. The heart's going to be pumping from that left ventricle and providing quite a thrust. And then veins are going to have a thicker tunica externa. Generally speaking, they also have a larger lumen. They tend to be less elastic and less collagen fibers than the arteries. And then these capillaries, they only contain the tunica intima. That is, they do not possess a subendothelial layer. Let's now take a closer look at arteries. In the upper right, we see a histology image showing those elastic fibers that are interspersed between the walls of that blood vessel. Again, that idea of absorbing that recoil. Let's start our discussion on arteries at the heart. So thinking about that left ventricle of that heart, giving a nice thorough squeeze, that blood comes shooting up to the aorta in that aortic arch. And then after that, these arteries are then going to kind of branch into smaller and smaller vessels, decreasing as they go along. They're going to decrease the diameter of the lumen, and also as we go along away from the heart, we're going to decrease the amount of elastic fibers, but we're going to increase the amount of smooth muscle as the further away we go from the heart. So we see that there's three general categories for arteries. We can classify these into three types. That is, elastic arteries, muscular arteries, and arterioles. The elastic arteries are going to be the largest of the arteries, and these have diameters roughly from 2.5 to 1 centimeter in width. These elastic arteries are called this because they tend to stretch to accommodate the blood when that left ventricle ejects blood during systole. So this recoil then propels the blood during diastole. Our second type, the muscular arteries, generally speaking have diameters from 1 centimeter to roughly 0.3 millimeters. The elastic fibers and muscular arteries are going to be in two layers. We have an internal elastic lamina, and then we have an external elastic lamina. And the idea behind here are we are going to sort of distribute a major and larger network of arteries to where they need to go, which tissues do they need to perfuse. And then we arrive at the arterioles, the smallest in diameter. And these are going to range from 0.3 millimeters to 10 micrometers. These arterioles are often in a state of sort of partial contraction. You might call that vasomotor tone. Therefore, they have a significant role in maintaining blood pressure. And then from these arterioles, later on, we'll see how capillaries and capillary beds come from these arteries. In general, these blood vessels are bringing oxygenated blood towards the tissues they need to perfuse. And this is a nice summary showing blood vessels compared. We get to compare and contrast those elastic muscular arteries with the arteriole. Notice how we compare and contrast the elastic tissue, the smooth muscle, and the prevalence of the fibrous tissues. And we arrive at capillaries and capillary beds. Continuing on with that same chart as the previous slide, notice how things like elastic tissues, smooth muscle, and fibrous tissues are gone. Again, the endothelium is continuous from the arterioles. So at these capillaries, this is where we're going to exchange substances from blood into the tissues. And these are going to be shorter vessels and smaller in diameter. When we say smaller in diameter, we're talking roughly 8 to 10 micrometers in diameter, and the length is on average about 1 millimeter in length. This narrow vessel diameter means that erythrocytes must travel in a single file line through each capillary. And again, because of the nature of the tissue and because of the diameter of this capillary, it makes it an ideal exchange site for substances between the body and the blood. I'm unsure if this movie link at the bottom works, but you could give that a shot if you'd like to. Let's take a closer look now at 3 types of capillaries. If one were to make some observations about the images in the right-hand corner, they might notice some differences in the cell wall. The blood vessel wall, I should say. That is going to be the difference between these 3 types. So let's look at our first type, the continuous type of capillary. This is marked by a complete lining. And so in this type of capillary, we have endothelial cells that have a continuous surrounding around the lumen. These endothelial cells are joined together by what's called tight junctions. And then we still have to get materials in and out of that vessel wall. So we have gaps in there called intercellular clefts. This is a sort of gap by which materials can pass through. And we're going to find these continuous capillaries in muscle, skin, lungs, and the central nervous system. Our second type of capillary is going to be fenestrated. Again, these are composed of complete endothelial lining around the lumen, but we're going to have small regions of fenestrations. These fenestrations are small pores, so to speak. And these pores are going to allow larger molecules to pass through. So, for example, we might find fenestrated capillaries in the small intestines, the ciliary process of the eye, the choroid plexus, or the kidney. And then the sinusoidal capillaries are an incomplete lining. So in other words, the endothelial cells are not tightly joined together. We have large openings or gaps that are going to allow for the transport of large substances between blood and tissues. And we might find sinusoids in bone marrow, liver, spleen, and in some endocrine glands. Again, the main idea of capillaries is it's going to be a site of exchange of substances. Let's talk a little bit about capillary beds. In the upper right is a nice histology image showing the red blood cells squeezing through that capillary in single file lines, so to speak, in rouleau. On the outside is going to be the nuclei of the endothelial cells. So let's look at microcirculation. In other words, a smaller circulation pattern within these capillaries. We have two types occurring here. We have true capillaries, and then we're going to have vascular shunts. And these true capillaries are going to be a network of capillaries, and they all come attached to a meta-arteriole. And so think about a normal daily activity where we might perfuse tissue in the far extreme of our digits, our fingers, and the tips of our fingers. If you give them a press right now, you might press them briefly, let go. You see the yellow turns to red. That's from the perfusion of that tissue. But let's look in a situation possibly where that red did not come back. Let's say, for example, frostbite. The human body needs to conserve that blood, that heat, and that warmth. So how do we close off that circulation? How do we cut off blood flow to certain more important areas? We might produce a vascular shunt. So that's shown in step 2. Step 2, or item 2, is showing a vascular shunt, and that is the network is sort of cut off, so to speak. And this way the blood travels from the arteriole to the venule directly through a meta-arteriole. And that's done thanks to these pre-capillary sphincters. If you look closely, there are tiny capillary sphincters that open and close and allow blood to be either cut off or flow through. So in the example of frostbite or frozen fingers, we might get frostbite on the fingertips because of loss and cutoff to our blood flow, but our main organs in our chest cavity will remain intact because we have conserved blood flow to the more important regions. Well, we've talked about arteries, we've talked about capillaries, and let's now turn our attention to veins and venules. As we discussed earlier, these veins are going to bring deoxygenated blood, generally speaking, back towards the heart. So these veins have the function of draining the capillaries, and they're going to be larger. So these veins are going to be larger than venules. The venules are going to be small in diameter and a little bit leaky, and the veins are going to be larger in diameter with a little bit of tunica media. These veins are going to return blood back to the heart, and interestingly, they can act as a blood reservoir. Now, keeping track with our diagram we've been talking about, notice how the endothelium is always present, but now we're starting to add elastic tissues back in, and smooth muscle is returning, as well as an outer fibrous tissue. So these veins then merge, again, starting with venules. It would go capillaries, venules, and then veins. So these veins are going to merge into larger and larger vessels, and as we merge together, we're going to get a larger lumen diameter as they approach the heart. And these venules, on average, are about 8 to 100 micrometers in diameter. Roughly speaking, it changes names. A venule then becomes a vein when its diameter is greater than 100 micrometers. This diagram shows how blood might make it back to the heart. So most veins contain valves, and these valves prevent backflow of blood. Again, we want blood moving in a one-way direction in this system. So also with these valves moving blood continuously, that's going to prevent blood from pooling in the limbs, sort of staying there together. It is interesting to note that the largest percentage of blood within the systemic circulation is found within the veins. So therefore, veins can function as blood reservoirs. Also, something noteworthy in this picture is the presence of skeletal muscle. We see that the veins in this diagram lie between skeletal muscle groups, and therefore movement and exercise might help the movement of blood returning back to the heart, since these veins do not have the high pressure of the left ventricle behind it. They need a little bit of assistance getting back in there. Here's a general overview of veins in the human body. As mentioned earlier, systemic veins do act as a sort of storage reservoir. And we also use the term sinuses when we talk about the venous system as well. So for example, the heart has a coronary sinus. So in terms of a blood vessel, a sinus is a vein that has a very thin wall and no smooth muscle to alter its diameters. Another example would be the sagittal sinus in the brain, or the skull, I should say. On Looker's right is a general map of all the vessels in the human body. For our lecture class, we're not memorizing that structures. That is mostly for the lab class to go through those venous structures. We turn our attention to a homeostatic imbalance of veins, and that is varicose veins. These are going to be twisted and dilated superficial veins. As one can see on Looker's right, these are prevalent. They come out through the dermis and the epidermis. Typically, these varicose veins are caused by the valve malfunctions. So we get leaky, so to speak, venous valves. And this could be from a congenital defect inherited genetically, or it could be because physically induced because of prolonged standing or perhaps pregnancy. And what we're seeing in this diagram, or I should say this image, is pooling of blood. So the lack of valve function causes blood to pool in one area. The vein swells and it bulges. So extra pressure is going to force fluid into the surrounding tissues, and that's why we see the nearby tissue being inflamed and tender. Typical sites for varicose veins: esophagus, lower limbs where it's hard to pump blood back to the heart. Also in the anal region. In the anal rectal region, these varicose veins are more commonly called hemorrhoids. The deeper veins are not susceptible to varicose veins because we have the support of surrounding muscles. So these are occurring mostly on superficial veins. Treatment include sclerotherapy, radiofrequency, occlusion, surgical stripping. These are all possible treatments for varicose veins. Let's jump off of veins for a little bit and jump back into arteries for a few. We see that now we talk about a special type of arrangement of arteries. And if we looked at a map of blood vessels in the human body, we would see that blood vessels are mostly arranged in a simple pathway. But later on we'll talk about a sort of alternative pathway. When we say a simple pathway, that might be something like one artery, one capillary bed, and a vein associated with the organ or the body region. So take, for example, a renal artery is going towards the kidney. So we might use the term end artery, an artery that provides only one pathway through which blood that can reach an organ. Well, we have sometimes alternative circulatory pathways, and examples include anastomoses and portal systems. And so here we see an anastomosis is going to be a joining together of blood vessels, the union of two or more arteries. The reason for this is that blockage of one of the pathway has no effects. There is a quote-unquote detour, so to speak. So for example, we see that the circle of Willis in the human brain is a good example of this alternative pathway. So again, if we had blockage in one of these arteries here, there would be a detour so that all the tissue in the human brain can be served with perfusion. So here's a nice overview of vessels we have discussed and vessels that we are just about to discuss. So let's start up here at the heart, and we'll move clockwise. Recall that this heart is going to have oxygenated blood coming out of the left ventricle. We move clockwise into elastic arteries. Again, they're elastic because they can absorb the recoil effect of that left ventricle. Those are going to conduct vessels to our muscular arteries. We call these distributing arteries because they distribute the blood to the organs they need to go to. That then gets us to the arterioles, the quote-unquote resistance vessels. This is where we're going to then transfer that blood to the meta-arterioles that then bring the blood to the capillaries, the aka exchange vessels. Notice in this diagram we have sinusoids there at the capillary. Again, the idea is that we're exchanging important substances to the tissues that surround it. From that meta-arteriole, we're going to jump into a venule, and that venule is then going to bring that deoxygenated blood back to smaller veins, and smaller veins are going to increase in diameter, turning into large veins. We sometimes see a sinus along the way, and then we come back to the heart for another pump around, possibly to the pulmonary circuit where we pick up the oxygen, and then back into the left ventricle to repeat the process. Notice one thing we did skip over is the area in green, and that's called the lymphatic system. So again, we're bringing fluid to these capillaries. Well, let's look at the bottom in purple. Not all fluid that exits the capillaries through filtration gets reabsorbed. It turns out that some of it stays actually in that tissue. So the lymphatic system is then responsible for returning excess fluid back into the bloodstream. And shown in green are lymph vessels. These lymph vessels reabsorb excess fluid, they filter it, and then return it into the venous circulation. So next week we talk about these larger things called lymph nodes where that filtration occurs. Let's now turn our attention to a homeostatic imbalance of these blood vessels, arteriosclerosis, more commonly called presently atherosclerosis. And this is going to be a hardening of the artery walls. The walls of the artery become sort of thick and stiff. And this atherosclerosis is characterized by the presence of an atheroma. And that atheroma is going to lead to thickening of the tunica intima, and therefore a narrowing of the arterial lumen. And that can be viewed in the above images showing how the lumen is substantially pinched off. We get common sites for these homeostatic imbalances include the aorta, the coronary, and the carotid arteries. Heart disease is a giant problem for medical providers, both in the past, present, and the future. It's a large issue that many future nurses will be taking care of. So let's talk about some of the risk factors for atherosclerosis. These include things like high blood pressure, smoking, diabetes, lack of exercise, obesity, high blood cholesterol, poor diet, and a high amount of genetic history in heart disease can all attribute to atherosclerosis. Roughly speaking, about half of the cases are going to be linked to genetics, and then smoking and obesity are the next two largest risk factors to blame for atherosclerosis. And so what might be happening in these artery walls are cholesterol particles that build up. And so we get a buildup of a sort of plaque. And this plaque, or these cholesterol particles, can come from two main sources: from the foods we digest or lipids formed by the liver. So as we mentioned earlier, a high blood cholesterol level is a risk factor for developing heart disease. Let's take a closer look at some of these lipids that are transported in the form of lipoprotein. So these lipoproteins are going to be particles in the blood that carry items like cholesterol, they carry fats, and they carry proteins. And again, there's a certain genetic component to how much your body makes of these lipoproteins. Let's look at a couple varieties. We have VLDLs, very low density lipoproteins, and these are going to cause an increase in fatty plaque formation. The next type are LDLs, the low-density lipoproteins, and these are going to be associated with the formation of fatty plaques. And then HDLs, or high-density lipoproteins, are going to remove excess cholesterol from circulation. So since getting a high amount of cholesterol is a sort of bad thing for the human body, let's look at ways that we might be able to mitigate that response. Just want to cross this out since that has been updated. So one way we can sort of bring our cholesterol levels down to a reasonable level is to eat a healthy diet. People with a high number of lipoproteins do appear to benefit from a diet of good fats in their diet. So when we say good fats, we're talking about nuts, fish, avocado, olive oil, things they sometimes call a quote-unquote Mediterranean diet. So we can see on this slide we have certain levels of cholesterol we want to look for, certain levels of LDLs and HDLs that we're shooting for. Let's see some other ideas that we might be able to reduce blood cholesterol levels. Exercise. Perhaps 30 to 60 minutes of moderately vigorous physical exercise every day will help keep our blood cholesterol in good levels. Another idea is to stop smoking. Quitting cigarette smoking is a good choice to reduce blood cholesterol levels. Controlling your weight is a good choice. Cutting off alcohol is an excellent choice. And if those things do not work through diet and exercise and lifestyle habits, we can put in certain medicines. So for example, we have pharmaceutical agents called statins, and these statins are going to help lower the cholesterol levels, but they do not help remove the plaques. So that does it for part 1 of blood vessels. Come back around, we're going to do part 2 of the blood vessels, and that's going to be loaded up shortly.